we will see what is cell signaling. First we must have to know about so today our topic is cell signaling. First we must have to know about the ligands. What are ligands? Like we have many hormones, we have drugs,
if the receptor is present at the surface, and in the next case you have the cell and the receptor is present within the cell. How is it possible? First, those ligands having higher molecular weight, for example, we have peptides. Peptides have higher molecular weight, so their receptors will be present at the surface. Second thing, they are highly polar. That means they are chargeable. They have highly charged.
so they are highly polar. For example, we have catecholamines, and in the catecholamines, we have epinephrine, norepinephrine, and dopamine. And here, this is also called intercellular. In this, the receptors are present within the cell, and the property should be they must have low molecular weight. Low molecular weight. For example, we have T3 and T4, and we have also one more example, retinoids. Second thing, when we talk here about this, they are highly lipid soluble. They must have highly lipid soluble. They are highly lipid soluble. As you know about the steroids. In the steroids, we have progesterone, we have estrogen, we have testosterone, aldosterone, we have vitamin D, we have glucocorticoids. And this was all about the two types. Now we will explain them in briefly what is intercellular cell signaling and what is extracellular cell signaling. First, we will talk about the intercellular cell signaling. In this, we have first must have known about the receptors. In this surface receptors, we have mainly three types of surface receptors. Like I will make here first one. This is the cell receptor. For example, this is the receptor, and this cell receptor passes the membrane 7 times. 1, 2, 3, 4, 5, 6, 7. This is the receptor outside. The ligand will bind here. For example, this is the ligand. This will bind here at the extracellular domain, and this receptor has intercellular domain also. This passes the membrane 7 times, so we call this as 7-pass receptor. We call this as 7-pass receptor, or one more name, as you see it is just like a snack, so we also call this as serpentine receptor. One more name is for this is serpentine receptor. This is one type. Second type, we have, for example, this is the cell, and here this is the ligand binding domain, and this ligand binding domain, it will pass through the cell only once. It has also inner this intracellular this domain, and but it passes only once through the cell, so we call this as one pass receptor. We call this as one pass receptor, or the second name for this is enzyme receptor. This is the second type. Now we have third type of receptor. These all you know that these are the ion channels. This any ion channel, for example calcium or chlorine, will come inside or it will go outside. Now we will study these receptors briefly. These are all surface receptors. Now first we will talk about the serpentine receptors. In the serpentine receptors, we will also talk about the GI. As you all know, the G protein. So we will talk about the G stimulating, G inhibiting, and the G2 system here. First, I will make a cell. For example, this is the cell, and here I have, this is the receptor, passes it 1, 2, 3, 4, 5, 6, 7 times. This is receptor. If I am talking about here in the myocardial cells, for example, this is myocardial cell, and in this myocardial cell, this is the receptor. This receptor is beta 1 adrenergic receptor. Now any ligand, like we have growth hormones, or we have, for example, I am saying that epinephrine binds here. This is epinephrine. Epinephrine binds to this ligand. Now the intercellular domain, it will pass this message to G protein. In the G protein, we have 3 peptide chains. First one is alpha, second one is beta, and the third one we have is gamma. But this G protein, we have with this has the energy of GDP. It has very low energy. As you know that the guy having very low money, he has more friends. That is, it is having less energy. So he makes the friendship with these two. But when this gets the information from the outside, this GDP comes out from here, it gets again with the GTP. Now he gets more energy. This alpha peptide will come out with this GTP energy, and when it comes out, this is all stimulating. We are talking about here stimulating G protein. And one more thing, all the receptors, all the serpentine receptors which works with the G protein, they are called as G protein coupled receptors. When this alpha with the GTP, it gives this information to adenylin cyclase. This is adenylin cyclase. What adenylin cyclase will do? After getting this information from alpha, it has one intrinsic protein here. Intrinsic protein. After getting this information, this phosphate information, when it gets this energy, it becomes active and it converts the ATP to cyclic AMP. That is called CAMP. Now one more thing, here this is the ligand, it acts as the first messenger, it gives the message. Here now this message gets transferred into the cyclic AMP. This is also called as secondary messenger. Now when CAMP gets energy, its quantity increases here. It is also called cyclic AMP, is also called secondary messenger. Now when it will increase, this energy will go again to phosphate kinase A. Why I take A? Because I have adenine cyclase. This gives this secondary messenger will give this information to phosphokinase A. After that, there will be, now we will see one more thing here. Just we are talking that there are 3 steps in cell signaling. First one is reception. That means ligand binding with the receptor. That is reception. Second thing is transduction. The information or the activation, or we can say the energy which the receptor gets, it gets transferred from different proteins. This is called transduction. Now we will get our response. Whatever is the response of this ligand. First, if this energy, if this gets converted, for example, if this is any enzyme, if it gets the phosphorylation of this enzyme will come into the cytoplasm and it will alter the metabolism. It will alter the metabolism. This is the first response. Second response, if it goes to the nucleus, it will get any transcriptional factor and it will alter the genome. Third thing, if this protein kinase A through ion channels, this was first response, this was second, and through third response, if through the ion channels it comes outside the cell, it will affect the neighboring cells. This was the third response. Now one more thing, this was stimulatory. We add here one more thing, like if there is acetylcholine, as you all know, it acts as the inhibitor. This will bind this, for example here I have again this 7-pass receptor, but here I will make it this above. This is here, when this acetylcholine binds here, this will give this activation energy to G protein. Again we have alpha, beta, gamma, but this time alpha, beta, gamma, they are inhibitory. Here they are stimulating, but here they are inhibitory. All the 3 are inhibitory. This alpha inhibitory will come and it will deactivate this adenine cyclase. This is the inhibitory. This was S system, this was I system. So there will not be any alter in genome or any alter in metabolism if there is this inhibitory G protein. Now there remains 2. This beta 1 and beta I and gamma I. They come to here, this membrane, and there is a ion channel. What they do, they take this potassium from the cell to the outside. This is again to alter the metabolism of the cell. This was one system. Now we will talk about, first we will talk about some medical cases. Like in medical science, let me tell you about the first one. We have pertussis. Pertussis, it is caused by Bordetella pertussis. What happened? This pertussis give a toxin, and this toxin binds with the receptor and it comes inside to the cell. Here we have G protein. Inside the cell we have G protein with alpha, beta, gamma polypeptides. What it happens, this toxin will come outside and it binds with them. This alpha cannot go outside from the G protein, so this will become off. This we can say this is off. That is off forever. Off is off forever. When it gets off, there will be pertussis, or we can also call this as whooping cough. This name Bordetella pertussis, bacteria. Second case we have Vibrio chloria. It is chloria caused by the Vibrio chloria. This Vibrio chloria, what will happen here? Again, the toxin will come to the G protein. This is G protein, and when it binds with the adenine cyclase here, this is adenine cyclase. What will happen? Right from here, alpha will come, it is beta, and this is gamma. Alpha with the energy of GTP, with very high energy, it comes to here. it comes to here with GTP. Now what can happen? Its intrinsic domain can break this GTP, but it cannot break this energy. It will remain always attached to this, and there is always formation of secondary messenger. When the secondary messenger increases, there will be a disease called Vibrio cholera, and this remains on forever. So we also call this as on is on forever. This is the second name for it. So these are the two medical cases which we have studied in cell signaling. After that, about the GQ system. One more system we have, GQ system. What is GQ system? Just like we have first GI, inhibitory system, yeah, G stimulating system, same is the case with GQ system. Like again we will take a cell. What will happen? Here is again we have, but this case GQ system, we cannot study this in myocardial cells. We study this system in smooth muscle cells. Smooth muscle cells. When this ligand, again I will say this, when the ligand, this ligand epinephrine binds with the receptor, when this ligand binds with the receptor, this here we have first one we have receptor beta 1 adrenergic receptor, but here we have receptor alpha adrenergic receptor. When this ligand binds to this receptor, its G protein gets signal. It is having alpha, beta, gamma. But here you must have to know that here the alpha, beta, gamma, it should be alpha Q, beta Q, and gamma Q. Again, it is having an energy of GDP. But when it gets the energy of GDP, it comes out from these two friends, alpha Q with the energy of GTP. It goes to one more protein, which we call as phosphate lipase C, PLC. This is called phosphate lipase C. This phosphate lipase C having intrinsic, like I am making, this is the very dangerous thing. Here you must have this will do. Here in the membrane we have PIP2, phosphatidyl inositol, 4,5 bis phosphate. It will cut this, it will break this into IP3, inositol triphosphate, and diacetyl glycerol. What will happen now? This IP3, inositol triphosphate, will come to the endoplasmic reticulum. This is endoplasmic reticulum having the pump on its surfaces. It is very rich in calcium. This will come to here, and through this pump it will come out from the calcium from the endoplasmic reticulum. And this calcium, when binds with the one more protein called calmodulin, when this calcium binds with the calmodulin, it forms a complex, calcium calmodulin complex, and this calcium calmodulin complex will give information to, you must have to see one thing, that if this is calcium, this is calmodulin, it will like give information to calcium calmodulin kinase. After that, the response will be same, like if it go to the cytoplasm, it will alter the metabolism. If it go to the nucleus, it will alter the genome. And through any ion channel, if it come outside, it will affect the nearby cells. And this diacylglycerol, this is also having like one more thing. Here we have phospholipase C. In the first case, we have adenylene cyclase, having adenylene cyclase, but here is C, so we must have to, there will be phosphate kinase C. Again, this phosphate kinase C having all the three responses. If it go to the, again, if it go to the cytoplasm, it will alter the metabolism. If it go to the nucleus, it will alter the genome. And if come out from any ion channel, it will affect the neighboring cells. This was the GQ system. Now after that we have some this is like this, these all were 7 serpentine receptors. Now we will talk about the enzyme link. Enzyme link receptors, or we call them as one pass receptors. one pass receptor. So in this case we must know that in this case we will talk about the tyrosine kinase. So here tyrosine kinase. For example, this is the tyrosine kinase having the ligand bind. This is the receptor. When the ligand binds, for example, the ligand binds here insulin. As you know that insulin is a growth hormone, or any endothelial epithelium growth hormone will bind to this receptor. When it binds to this receptor, what actually happens? This is having intercellular domain. What is happened in the intercellular domain? Having the intercellular domain, this intercellular domain is having tyrosine on its surface. It is having tyrosine. What it will do? Here it will do this. This is the phosphorylation. This information will given to sulfur hydral SH2, and after SH2, for example, this is the SH2. This gets the information. This SH2 will give this information to very interesting protein. This is the seventh protein. This is the 7. So we call this as son of 7 proteins. Son of 7 proteins. This son of 7 proteins will give information. Again, after taking the information from here, it will give this information to, again it will be same like from adenylene cyclase. If it gives the information to adenylene cyclase, from the adenylene cyclase, all the 3 proteins that will come, this the previous system we have, it will be same like for that also, this tyrosine kinase also. But one more system we must have to know. This is tyrosine system, but here in this system we have, this is tyrosine kinase, but this is associated receptor. Here we have tyrosine kinase receptor, but here we have tyrosine kinase associated receptor. Same here, growth hormones can bind growth hormones, or here it can, the ligand will be interleukins 2, or it can be prolactin. It can be prolactin, or it can be any cytokines, or it can be like T, that is T lymphocyte cell receptor, or it will be B lymphocyte cell receptor. When they bind to this tyrosine kinase associated receptor, what will happen? Here we have present JAK kinases, JAK, which we call JAK kinases. JAK kinases. Here we have presence of tyrosine. In this case, there was autophosphorylation of this tyrosine, but in this case there will be cross phosphorylation. When it becomes phosphorylated, this information will be provided to STAT protein, that is signal transducer activating transmission. When this gets this information, they become the dimer. They will make a dimer, and this dimer will come into the cell. For example, this is the cell and having a DNA. This will come into this cell. This dimer will come and it will act as a transcriptional factor. So there will be the formation of messenger RNA, and there will be the formation of proteins. And so these proteins, they can alter any metabolism.